Good afternoon, class. We are meeting today to revise the March paper free state that was written this morning. So we are just going to work on the question paper, and various answers or suitable answers are going to be provided as we discuss the paper through. You can see this is a free state paper, uh, March 2026. So actually, we are just going to mark, we are going to review, we are going to evaluate, and also just understand the paper that was written this March of 2026. It is quite good for us to understand and also make sure that Term 2 is loading in style. Without wasting any time, we always have instructions which are given there. I'm sure that grade 12, you know that instructions are quite important. You read them before answering any piece of work, and then we just go straight to the questions that are given here. It's multiple choice. I am always reminding and encouraging students that in option or answers or questions that have got options or a multiple choice, I don't advise and expect any one of you to leave blank spaces. You should always try to give the answer that you can think it is right, given that you don't know the answer, unlike to leave blank spaces. So first question it says, the inner lining of the uterus, that is richly supplied with the blood vessels. I'm sure that you remember, if you look on the female reproductive system, the endometrium, it is the one that is nourished, or that actually that is supplied with blood in order to prepare for implantation. So we understand that endometrium, it is richly supplied with blood. As we understand that, we also know the shedding, the monthly shedding, which is caused because of the breaking down of that endometrium as a result of failure of fertilization or pregnancy. So the answer is B, and the normal place where fertilization takes place in the woman, or in it is the fallopian tubes. We say that fertilization occurs in the oviduct. The fallopian tube is responsible uh, for fertilization. And I want you to understand again, quite a lot of times, I have seen that other question which comes in, whereby there is growth inside the uterus, inside the fallopian tube, that would affect and disturb or prevent the zygote to move into the uterus, which quite a lot of times we find out that it's a rare case anyway, but it does okay. The What do we call that one? We are saying ectopic pregnancy occurs when the fertilized egg fails to move out of the uterus, but fertilization, it occurs in the uterus, in the fallopian tube. The uterus is actually there as the womb. The uterus is the womb where the baby grows. It actually provides the housing conditions for the development of the baby. The nitrogenous base pairing in DNA was discovered. The discovery of nitrogen, uh, the nitrogenous base pairing, that it was discovered by Crick and Watson. Crick and Watson did discover this. I want you to understand that it's quite a common question in any paper, which you need to understand the discovery of DNA. Who discovered DNA? We are saying it was finally discovered, and it was actually the prize given to Crick and Watson. And then a short piece of DNA containing 21 nucleotides in each strand was analyzed. The number of some of the different nitrogenous bases in each strand is shown below. I'm quite aware that at grade 12, we know very well that DNA strand, it is a double helix. It has got two strands. It has got strand 1 and strand 2. So what we need to understand is to say we can say strand 1, we can say strand 2, And then what happens is that there is always complementary pairing of bases that I want you to understand. That when we look on the structure of DNA, bases always do pair complementarily to say the number of A must be equals to what? To T, and C must be equals to G. We have got weak hydrogen bonds that also forms in between there. The weak hydrogen bonds forms in between the complementary bases. I'm sure that we understand it. And then what I just want to remind and make it clear to you is that we have got base pairing, C pairs with what? With G, and A pairs with what? With T. And what happens is that if we have got this tells us that total number of A is equally the same as number of what? Of T. We get A, we must get T. Each strand, the number of nitrogen bases in strand 1 must be equal to the number of we are saying if we have got C nitrogen, if we have got cytosine bases that are 8, it must also mean that in other the opposite strand we must also have 8 what? We must also have 8 guanine bases. If it happens that we have got four adenine bases, it should also mean that we have got also four guanine bases. That is according to say the base pairing rule. The base pairing rule to say strand 1 must say we have got A here. We therefore should have T there. We have C, we have G, we have G, we have C, we have T, we have A. We have got A, we have T, we have C, we have G, we have G, we have C. So basically what I'm saying is the base pairing rule explains that the total number of nitrogen bases which are for complementary bases must be equal. That's what we need to understand. So from this question that we are given here, it has to explain that we are given the number of nitrogen bases that are given on strand 2. Strand 2, it has got, we are saying we have got the total number of bases, which we say that if, for example, we say the number of nitrogen bases, let's say the number of cytosine bases, cytosine on strand 1, we are saying strand 1, it should be equals to G on strand 2 in terms of the base pairing rule. But anyway, let's see, we are saying strand 1, it has got cytosine bases 80, and then it means therefore that the we have got the G here, we have got G in terms of the other strand to say strand 2, it has that. So what we need to understand is that the basis must be what? Must be complementary, which therefore means to say G80 is equals to also 8. But in any case, in this option that we are given here, we don't have such an answer. But I want us to understand that we are told that there are 21 nucleotide bases. It is given that there are 21 nucleotide bases. So I think this must be calculated also as to say that G in strand 2, say G, we say that G in strand 2 is equals to, we should say that we must say it is equals to 21, which is the total number of nitrogen bases is equals to 21, the total number of nitrogen bases that we have there, which we can say that 21 minus the sum of bases that are present. So 21 minus, we say that we have got 4A minus 4 plus 6 plus 8 minus 4 plus 6 plus 80, which we might otherwise say that 21 minus 18, 21 minus 18, which can otherwise give us to say we get what? We get 3. And then 3, we can say G in strand 2 is equals to 3. But I'm saying the the exact method that I want us to understand is that we must otherwise work on the best pairing rules between strands. The best pairing rules between strands, I want to accept and believe that that is the best answer that must give us this answer. But from here, the best pairing rule does not give us 8, which is again, which does not appear on the options. So for now, we can just say that we can infer to say that the answer can be what? Can be B, which we can say otherwise B there. But the best pairing rule must give us an 8 in that respect. And then give the correct biological term for each of the following descriptions. The correct biological term for each of the following descriptions. So now, a reproductive strategy where the development of the fetus occurs inside the uterus of the mother. We have talked about this again on Metric Hub. It is given and explained enough that we say that this is uh, I'm saying this is vivipary, which allows the fetus to actually develop inside the uterus of the mother. We are saying the vivipary, it is actually the embryo or fetus develop inside the body of the mother, eventually leading to live birth. So it is what? Vivipary. It is vivipary. And then the type of fertilization in which the nucleus of the sperm fuse with the uterus of the ovum inside the body of the female. We said there are types of fertilization. It is internal fertilization, which explains that there is penetration and there is deposition of the sperm cell inside the reproductive organ of the female, and therefore the sperm cell moves and facilitate fertilization inside inside the body of the female. It is called internal. Where it is outside, it's called external fertilization. And then all chromosomes except gonosomes, we are saying gonosomes are also called the other term. These are also called we are saying these are also called, we are saying sperm cells except the gonosomes. The gonosomes, we are saying these are also called sex cells. So except these ones, the other term for others are number 1 to 22. Sometimes they can say 1 to 22 chromosomes. They are called autosomes. They are called autosomes. That is, I'm saying, is number 1, which is number 1 to 22 chromosomes. They are called autosomes. And then 23, we say that 23 is called the gonosome, which we are saying it is the XY, it is the gender, it's the sex chromosomes or sex cells. We say that these are sex chromosomes, which we say that I want you to understand again that the X chromosomes, we say that they are long, and then we have got that is equals to say it's a what? It's a female. I want you to note that. And then we also have situations where we have got this one, which would say that we have got a long and short, which is equals to male. I want you to understand that there are such situations where you are asked to say that what is the gender given the karyotype. So you need to know that if we have got a long and short chromosome, it's a male, and then long alone or long, it's a female. It's the building blocks of nucleic acids. We say that nucleic acids are built up of nucleotides. They are built up of what? nucleotides, nucleotides, which we said that we have got nucleotides, nucleotides, which we say that it has got the sugar, the deoxyribose sugar, it has got the deoxyribose sugar, it has got the phosphate and nitrogen base. We have got uh we have got the P, we have got the sugar, we have got the nitrogen base. It is called a nucleotide. That is called a nucleotide. So this is called the nucleotide, which is a building block of what? Of nucleic acids. And I want you to understand that nucleic acids, we have got what? RNA and DNA. And then born with eyes closed. We talked about forms of development. We are saying the forms of development, we have got precocial and atricial development. So we are saying they are born with eyes open, with eyes closed. I'm saying eyes closed, it is precocial development, which is A alone. When the eyes are closed, we say that is a precocial development. And then atricial, the opposite of this one is what is atricial, which are born with eyes open. With eyes open, we say that is what? That is atricial, atricial development, which is another question which would come on a certain other time. But I just want you to know that. Production of haploid gametes, we are saying that haploid gametes are produced. We are saying this is half, these are these are called any gametes, or we are saying sex cells. Any are produced by what? Which is half. Half is produced by what? By meiosis. And then mitosis, it maintains the same number of what? The same number of chromosomes. So it is meiosis which causes haploid gametes. It is meiosis which is B only. And then structure that holds the two chromatids together. The two chromatids are held together. We are saying they are held by the centromere. The centromere holds the two chromosomes to not two chromatids together, which is also B. I said that we have got a chromatide, and then we have that piece, and we have got a chromatide. The piece that is holding the two chromatids together, we call that, this is called a what? A centromere. The centromere, the piece that holds two chromatids together to form a chromosome. Also, please refer to metric hub. There is quite a lot of explanation which has been given in terms of the scientific terminology in genetics and inheritance, and also DNA in general. So diagram represent the type of terrestrial, the type of egg in terrestrial vertebrates. We say the terrestrial, these are, we are saying animals that are living on land. These are organisms that live on land. We call them terrestrial. And then we have to answer the questions that are there. This is an amniotic egg in basic way, in general, in its scientific term. Amniotic egg, I also have explained enough on the channel. Please check on Metric Hub. There is quite a good explanation and lecture explaining an amniotic egg and every other relative question that comes with it. So this is called an amniotic egg. Amniotic egg is called an amniotic egg. So I'm saying that it is a characteristic of an egg that comes from terrestrial vertebrates, which we can give, such as reptiles. We can talk about birds, and we can talk about other mammals, and contains extra embryonic membranes that protects and nourish the what? The embryo. So as you can see, there are many membranes that are there, which we can say that they are working to protect and nourish the what? The developing embryo there. And then identify Identify E. E is amnion. E is the amnion. So what you can say is that it is actually encloses the amniotic cavity. It is the one that houses the amniotic cavity, or it says it houses the amniotic fluid, which I want you to understand that it protects the embryo from mechanical shocks and dehydration, and also the flexes in the development of the fetus or the developing embryo. So the amniotic cavity is contained in E. E contains the amniotic, it is the amnion which contains the amniotic fluid. And then give the letter and name of the part that prevents dehydration and protects and provides protection. The one that prevents dehydration, we are saying the party that actually that prevents dehydration, we are saying, I'm sure that you should know that it's more or less the same from where we are saying. We are saying dehydration, it is because of the amniotic fluid. So it is 1.43. Give the letter in the name. The letter in the name. We are saying it is the amnion. So it is the amnion is letter E, and also it is the what? The amnion. The amnion again. It is kept on being asked. It is E, which is the amnion as well. So the answer is what? It is E, amnion, and it is letter E. And then store the metabolic waste from the embryo. The waste is actually, it is actually contained. It is actually from the what? It's from the alliantos. Alliantos actually, it is the one that that actually ensures that the waste, it stores the metabolic waste from the embryo. So we can say that that is C. We are saying allantois, the allantois, it is actually working for that purpose. And then forensic investigators examined a break-in at a company where high-value goods were stolen. DNA samples were collected from a broken window at the scene. 3 suspects were taken to custody. DNA samples were obtained for analysis. The results of the DNA analysis are presented below. So we have got the DNA analysis, the crime scene samples. We have got our suspect 1, 2, and 3. So what question is saying is that it says name the technique used in criminal investigation. This is called, the technique used is DNA profiling. This is called DNA. It is called DNA profiling, which we also say that this is called also DNA fingerprinting. DNA fingerprinting is the same as DNA profiling, which I want you to understand that it is sometimes called gel electrophoresis. Gel electrophoresis. It can also be termed like that. Which one of the suspect was at the crime scene? We see that which one of the suspect was at the crime scene. We are saying we look at the closeness of the DNA at the crime scene to those that are there. So we look at the various samples that are there. I'm saying this is the DNA at the crime scene. This is the sample from the crime scene, this one. So we are saying suspects are these ones. So we look at the relative matching of these samples. So we have got this bar there. We don't have it there. We have this one here. We don't have, it's not there. We have got that one there. We don't have it here. It is down there. We have this one there. It matches there. We have got this one. It also doesn't match there, and there it matches. And again, there it is not. We have got this bar there. It matches that one. That one there, it goes with that. So we are saying the likeliness or the matching of the sample to the suspect defines who was at the what? At the crime scene. As you can see, we are saying the matching of the DNA profile or the DNA that is collected from the crime scene versus the suspect. So we are saying you look at the matching of those. Sometimes they might not closely match, but we'll look at the closest one to the one that is taken from the crime scene. So we can say that it is suspect number 2. Give one observable reason for that. We are saying the pattern of the DNA bands for the suspect 2 matches the pattern of the DNA from the crime scene. Number 3, it is the pattern of the DNA bands for suspect 2. DNA bands for suspect 2, it matches. It actually matches the pattern of the DNA from the crime scene. It matches the bands. It matches the bands or a pattern which is from the crime scene. That is what happens there. We are saying there is a match, as you see the way we were marking there. Name 2 other uses of this technique. Let's say name 2 other uses of this technique, which is shown in the diagram. We can talk about 1, identification of genetic diseases. Identification of genetic diseases can be identified through this process. And then we can talk about also paternity determining biological parents. Determining biological parents can also be determined through our DNA profiling, which we can see also the closeness of matching. We can also use it in terms of determining evolutionary relationships between organisms. Evolutionary relationships between organisms can also be determined through through DNA profiling. We say organ transplant can also be some of those, that is genetic defects and stuff. So these are the uses that we can actually find with regards to question 1.54. And then we have to read this context. Down syndrome, a genetic condition which leads to developmental delays. intellectual disability, physical characteristics. People with Down syndrome tend to have varying levels of learning difficulties, heart defects, and other medical conditions. Scientists at National Down Syndrome Society conducted a study to determine the effect of the age of mother of having a child with Down syndrome. So the procedure was as given here, and then maternal age incidence of Down syndrome 20 is 1 in a 2000, and then 35 1 in a 350, 45 1 in a 30. Those are the variations that are given there. And then what is the dependent variable in this investigation? Uh, we are saying determine the dependent variable in this investigation. We said that scientists conducted a study to determine the effect. We are saying the effect of mothers of the effect of age of the mothers of having children with Down syndrome. So we are saying uh, what is dependent variable? We are saying Down syndrome is dependent on the age, the effect of age of the mothers of the mother of having a child with Down syndrome. So children with Down syndrome, we are saying Down syndrome is dependent on age. Down syndrome is dependent on age. That is what is the it is actually uh the incidence of Down syndrome is the dependent variable. The incidence of Down syndrome, the incidence of Down syndrome is dependent on what? It is depending on age. And state two reasons why this investigation cannot be regarded as what? As reliable. Uh, Two reasons why this investigation can be regarded as reliable. We are saying for an investigation to give results which are confirming what is to be studied, we are saying that it can be based on various reasons. We are saying one, as you can see, we are told that the town were conducted out of 354,880 beds. So the larger the sample, the better the results. So we are saying one, large sample size, large sample size was used. Large sample size was used. And then we also want to say that it can also be, we are saying it can also be also good because we are saying it was used on, scientists used official birth statistics. Official statistics were used. So we can also say official statistics were used as another reason. Official statistics. Official statistics were used. It can also identify the fact that the results can also be reliable. And then next question is saying, list two characteristics of a person with Down syndrome according to the paragraph. We are given we are given the characteristics here. The Down syndrome is a genetic condition which leads to developmental delays, intellectual disability, and distinctive physical characteristics. People with Down syndrome tend to have varying levels of learning difficulties, heart defects, and other medical conditions. So we can give a list as much of this one. Developmental delays. We say developmental delays. Developmental delays. We can also talk about intellectual disabilities. intellectual disability. It can also form part of that. We have been talking about distinctive physical characteristics. Distinctive physical characteristics are also part of these characteristics that we are supposed to talk about. And then even learning difficulties, heart defects, they are listed in this. They are all various. We are saying they are given in the context. They are given in our uh, question or in our paragraph or infographic that is available there. And then calculate the percentage difference between having a baby with Down syndrome at the age of 20, Down syndrome of having at the age of 20 or at the age of 45. Show your workings and round off your answer to 2 decimal places. Calculate the percentage difference. Calculate the percentage difference having a baby with Down syndrome at the age of 20. At the age of 20, at the age of 20, we are saying it is given there that it is 1 out of 2,000. At the age of 20 is 1 over 2,000 multiplied by 100. Age of 20 is equals to 1 over 2,000 multiplied by 100. And then what we can get there, it can give us 0.05%. And then at the age of 45, at the age of 45, it is given also that age of 45 is 1 out of 30. 1 out of 30, so it is 1 over 30 multiplied by 100, which is going to give us 1 over 30. It's going to give us 3.33 percent. So we are saying the percentage difference is equals to 3.33 minus 0.05. 0.05%. If we minus that will give us which is equals to 3.3 minus 0.5. We get this gonna give us 3.28%. So we say the answer correct to 2 decimal place, which is correct to say it is correct. We are saying to 2 decimal place, that is 2 digits after the comma, which is 3.28%. Describe cause of Down syndrome in humans. The cause of Down syndrome in humans. What causes Down syndrome? They describe the causes of Down syndrome in children. This is a condition that is a disorder caused by non-separation or non-disjunction of chromosomes during Down syndrome. We are saying it is caused by non-separation they sometimes call it non-disjunction of chromosomes during meiosis. non-separation of chromosomes during meiosis, which can be either meiosis 1 or 2. So what happens there is that chromosome pair 21, it actually fails to separate during anaphase 1 or 2. Fails to separate during anaphase 1 or it can be 2, which I want you to know that as a result, one gamete or egg, as a result, what happens there? One gamete, which we are saying the gamete, we are talking about the sperm cell or the egg cell, actually receives 2 copies of chromosome 21. It receives 2 copies of chromosome 21. And then what happens there is that obviously the other will have none. So now what happens there is that if the gamete with 2 copies of chromosomes fuses with a normal gamete, the one with 2 copies, if a gamete with 2 copies fuses with, if it fuses with a normal gamete, if it fuses with a normal gamete, it results in which we are saying that is fertilization class that we are talking about. If fertilization occurs, if it fuses in fertilization, we are saying the resulting zygote, we have 3 copies of chromosome 21. The resultant zygote will have 3 copies of 21. It will have 3 copies of 21, which we are saying a condition known as trisomy condition. A condition known as trisomy. A condition known as trisomy, which we are saying results in 47 chromosomes instead of 46. And then therefore we are saying this condition, it is the one that causes what? It is the one that causes Down syndrome. So during meiosis, chromosomes may fail to separate, and then we have got a gamete that is formed with, which is going to be formed with 2N. 2N, as to say it is having 2N. So if it fuses with an N, if it fuses with an N gamete, it gives us that process of saying 2N. If we have got that 2N, to say that 2N, it fuses with N, it results in what? In 3N. That 3N is what we call trisomy. Is what we call trisomy condition. That 3N is what we call trisomy condition class. So that's what happens that we need to understand. We understand it that Down syndrome, it is a condition that is resultant from errors that occurs during gametes formation. The fact that we have got a gamete with 2 copies instead of one copy, and then it fuses with a normal gamete, which results in this illustration that I've shown here. And then we are given the next question to say, it's a diagram that represents male reproductive system. Give the letter and name of the structure that transports sperm cells to urethra. We are saying the letter and the name. So instead, we need to start with the name first. The name, we are saying the one that transports sperm cells to the urethra. It is the vas deferens, or the what? Or the sperm duct. So the vas deferens or the sperm duct, it is letter A, letter and name. So it is A, which is the sperm duct or vas deferens. It is letter A. The sperm duct or vas deferens is the one that carries, is A. It carries the sperm cells from the testicles to the urethra, and then gland that secretes an alkaline fluid that neutralizes the acidity in the vagina. We are saying it is secret. It is the neutralization is done by the prostate gland. So the prostate gland, it is represented by what? By letter C. Prostate gland, we are saying it is the prostate gland. It is C, which is the prostate gland. And then name the hormone that is responsible for the development of a deep voice in males. We are saying that this is caused because of it is caused because of the testosterone hormone. Testosterone. The testosterone hormone is the one that is responsible. Testosterone. It is the testosterone hormone. Testosterone hormone. It is the one for secondary characteristics development. And then during which stage of human development is the hormone produced? During which stage it is produced? During puberty. It is during puberty. Puberty is the stage of secondary characteristics development class. And then make a label diagram of the gamete produced. Make a label diagram of the gamete produced in part E. A label, we are saying draw a sperm. It is just asking to draw a sperm cell. We say draw a diagram of a label sperm cell. That's what you need to do. And then you just have to say question 2.24. 2.24. So we can say that the sperm cell, that's the sperm cell there. So we say it's an acrosome. We have the nucleus. We have got the head of the sperm cell. We have got the middle piece. The middle piece, I'm sure you know that we say that it contains the mitochondria. We have got the tail. The tail, the middle piece. which we say that it contains the mitochondria. That is the basic diagram that we can give there. A simple diagram like that. That's what we need to do. That's what we need to draw there. I think that's the basic diagram that we need to draw. And then, yeah. And then the next question is saying, describe the type of gametogenesis that occurs in females. They want you to talk about oogenesis. Describe oogenesis 2.3. They want you to describe oogenesis. That is, we are saying in female. That's what they want you to describe here. Describe the oogenesis. So what is happening is a process in which we are saying that it's a process in which gametes are produced, a process through which gametes are produced in female. In female reproductive system, we talk about oogenesis. So now what we need to understand is that they are produced in ovaries through meiosis. They are produced in ovaries through meiosis, which we are saying resulting in one large egg. It results in one large egg. One large egg cell is produced in smaller polar bodies, which degenerate in smaller polar bodies that dies. Smaller polar bodies, bodies that degenerate, they die, and then it begins before birthing. The process basically begins before birthing and is completed after puberty during menstrual cycle. That's what we can say. Begins before birthing, but it's completed after puberty during menstrual cycle. That's what we can say there in terms of this oogenesis process. And again, don't forget that it produces haploid. It produces haploid gametes. And then question 3 point, next question, it is, it wants us to say, state one place in humans where meiosis takes place. This is also a common question, which I always emphasize to my students, that you need to know where in plants and in animals do meiosis take place, whether in male and female. So we can say that testes, that is in male, and then we say in the ovaries in female, or we say ovaries in female. That's what we need to know. And then identify structure B. Identify structure B. Structure B, it actually looks at it's a chromosome. that's a chromosome. C, we said it's called a centriole. I'm sure I remember that is called the centriole. We have also said that when we go to meiosis, you should always be able to draw and label the structure of the cell. It's quite very important in order for us not to miss unnecessary marks. Give the function of structure A, uh, the structure A, which we are saying is the spindle fibers. We are saying A, it is the spindle fiber. So the spindle fiber, basically we are saying that it attaches the centromere of the chromosomes and contracts in order to move the position of chromatids or chromosomes. So in simple terms, we can say controls the position of chromosomes or chromatids during the process of meiosis. So what it does is through it by contracting, and what it can either contract or expand. I mean, the contraction and shortening of it pulls these, controlling the stages of what? Of meiosis. It actually controls the process of meiosis is controlled because of the spindle fiber, which actually contracts and expands to control these. You understand? That is what we need to understand in terms of this, and then identify the phase of cell division that is represented. This is actually, we can say, as you can see on the diagram, it is called anaphase. We can say that is anaphase 2. It is, we can say that is anaphase 2. And then the reason is because the sister chromatids are separating and moving to opposite poles of the cell. Separating and moving they are moving to opposite poles of the cell. They are moving to opposite poles of the cell. In other times, again, someone can argue to say that it can be also metaphase, because we have got chromosomes that are aligned at the equator. Someone can also argue to say that we can also say that it is metaphase because we have got chromosomes that are lined at the equator. That can also maybe match that one. And then question number next is saying, how many chromosomes will be found in a somatic cell of this organism? We are saying in this case, we need to know that the cell in the diagram, we say that it is in anaphase 2. So where chromosomes move to each pole, this is a haploid number, which is N. So we need to, first of all, to determine the number of chromosomes in the gamete. So in the number of chromosomes in the gamete, the cell, we are saying it has got, we can say that this can have 4. It can say that the cell has got 4 chromosomes moving to opposite poles. This is haploid number N. So N is equals to 4. So therefore, in order to determine the number of chromosomes in the somatic cell, it is 2N. So where N is the chromosomes in the somatic cell, and is twice the haploid number. So in this case, what we are saying is that this one, it must be known that we are saying number of chromosomes in the gamete, number of chromosomes in the gamete, we say is equals to, we are saying this should be known that we have got 4. It is 4. And then what happens there is that as we look at that, this is, which is also, we are saying this is haploid, which means it is half. So what we need to understand is that the number of chromosomes in a somatic cell, number in a somatic cell is equals also to, in the somatic cell, this is equals to, say, it must be 2n, which is also called diploid. So I'm sure that you remember to say 2n is where n is representing, where n represents the number of what, of cells. So it is 2 times 4, which is equals to what? Which is equals to 8. It is just a simple mathematics that we say 2n is equals to 2 times n, and then we are saying 2n is equals to, we are saying 2 times 4, and then 2n is equals to 8. That is how we can say this whole thing comes about. And then it says, name and describe the process in meiosis that contribute to genetic variation except one in the diagram. So genetic variation is also caused by, we can talk about another process of meiosis causing this. We can say crossing over. Crossing over, we say that it's exchange of genetic material. Exchange of genetic information between homologous chromosomes. And then what we are saying is that it is which occurs during prophase 1. Occurring during prophase 1. So now what happens is that it actually results in new combination of alleles on the chromosomes that increases genetic variation. So the crossing over process occurs by the chiasmata and it results in a new combination of alleles. It results in new combination of alleles, which increases genetic variation. That is what you need to understand when it comes to this. We can just go to the next question. The next question, it is just the diagram illustrate protein synthesis. This is quite a question which we have also answered severally also on Metric Hub. It has been answered also severally. For those who are following Metric Hub, you can see that these questions have been answered. They were answered extremely so and precisely correct. So we talk about the question that comes there is saying identify molecule A. Identify molecule A. The molecule A that we are seeing there, you can say that it is the DNA double helix. DNA double helix, which you can see there, it is given here that this structure is just, we are given this structure there. So that is called a double helix structure of DNA that is shown here. It's a DNA double helix, and then process B, it is transcription. It is transcription, which we are saying formation of what? Formation of mRNA. mRNA, it is called transcription. And then structure C, structure C, that is a peptide bond. It's a chain of amino acids joined together. This C is identifying the chain of amino acids that are joined together, that is a peptide bond, and then give the function of organelle D. It is actually responsible for, it is the ribosome. The question say give the function, not the name anyway. We are saying it is the ribosome. It is responsible for, it is a site of translation, a site for protein synthesis. Protein formation takes place on what? On structure D. That is where the reading of mRNA base sequence occurs, and then it assembles the amino acids into polypeptide chains, which we say the proteins, and then describe the process of translation. The process of translation, question 3.3. We can also have it. It has been also explained on Metric Hub. Explained also on Metric Hub. It is explained also on Metric Hub. What happens is that we are saying the mRNA is read by the ribosome in the specific order of what? Of its codons. The codons on mRNA are read through the ribosome, and then it is actually activated to send signal to tRNA that brings the corresponding anticodons to it, which would also bring the amino acids, and then the base pairing of the codons and anticodons is actually complementary. That actually means that A pairs with the U, C pairs with the G, and again, we need to understand that there are specific amino acids that are brought to the ribosome, and then peptide bond formation is formed as the ribosomes move along the mRNA. It actually, like to say, more tRNA molecules bring their specific amino acids there, which peptide bonds form between adjacent amino acids, creating a growing polypeptide chain, as we have seen there. So basically, that's the process that has to be understood. You can as well check it there. It is explained and has been marked quite clear that a translation protein synthesis, it has been explained fair enough, shows the triplets that form part of DNA molecule that codes for a specific protein. And we are asked questions which says, the first question is saying, give the anticodon of base triplet 1, the anticodon of base triplet 1, the anticodon of base triplet 1, we just have to check it. We said that the matching of the anticodon of base triplet 1, the anticodon of base triplet 1, that is the one that is given here. So what is happening is that this is base triplet. The DNA base sequence, we are saying base triplet on DNA is this one. So we are saying the anticodon, it comes where this, we are saying it comes, we need to understand that this, it has to come in a way that we need to understand the base pairing rules during transcription. DNA bases pairs with mRNA, that we need to understand. We are saying during transcription, DNA, we are saying DNA molecule, it pairs with RNA. So I can just maybe want to say that that is the structure where we are saying it is our DNA here. Our DNA, it has got, it has TCA, and then we must know that it matches with what? It matches with it matches with mRNA. So we have got our mRNA, which must have the complementary bases that pairs to it there. So here we say that T pairs with A, and C pairs with G, A pairs with what? With U. This is mRNA, this one. So now we are saying the anticodons, we must derive them from what? From this, to say that now we must have the base sequence which comes from the tRNA. We must have our tRNA. This is our tRNA, this one. That's our tRNA. So now we say A, it must have our, it has to have, it's what? It has to have, it has to have U. We have got U there. We have C. We have A. These ones are called anticodons. And these ones, the red ones, are called, the black ones, these ones are called codons. So what we need to understand is that there is a formula of understanding the sequence of these to say that mRNA is the one that you have got the codon. So you need to derive the codons first in order for you to get the anticodons. That's the basic understanding which a learner must have in order for you not to make mistakes. So in this case, we can say that the question is saying the anticodons of the base triplet, that one, it has to be known that it is the ones that we have calculated, that we said naturally base pairing, we say A, it pairs with U, C, it pairs with what? With the G. That is the natural base pairing sequence. So now, in terms of this, so we are saying our base triplet 1, our base triplet 1, we are given that it is what? It is given that our base triplet 1, it's given that is TCA. TCA that is given there, it is TCA, TCA, that is the base triplet number 1, TCA. So having TCA, so we are saying the we must give it like the way I did already have worked out to say we must give the mRNA, which we said that it must give us our mRNA, which is going to be saying it is AG. We said it is supposed to give us AGU. These are the what? These are codons. And then its anticodons now, we must bring the what? The tRNA. So we should have UCA. These are anticodons. So UCA should be the answer there. UCA. That's the answer there. UCA, according to the base pairing that we have calculated there. And then the correct sequence of amino acids from left to right. The correct sequence of amino acids from left to right. So we can go there again from left to right. We can just take them there. The correct sequence of amino acids from left to right. So we are saying the amino acids that we have got, we are given this. We need to identify the amino acids that should be coded, and then we identify them from the table here. So using the provided table, we are saying AGU, and we have got to give these we have to give their codons here. So the codons for this one, we must give the codons for all of them. So we are saying here it is A G U. On this one is A U C. On this one it is G G U. So we are saying these are our codons here. So we look for the codons. These codons, we identify their amino acids here, class. So AGU is arginine. This is the first one. And then we go also AUC, it is number 2. And then GGU is number 3. So we are saying, we are just mentioning them in their order as they are here, 1, 2, 1, 2, 3, which is called arginine, as isoleucine, and glycerine. That is what we need to give here. That is what we should give here. We're saying arginine. Our arginine, we have got arginine, we have got isoleucine, and then we have got the last one, which is glycine. I think, class, you have seen what is happening there. I said you calculate the codons for this base triplet that you are given here. So our conditions are calculated. Our amino acids are calculated on they are calculated on these red codons. These are the codons that we are using. We use these mRNA codons. This is the mRNA codons that we should use in order for us to get these answers. You look for AGU, AUC, and GGU. Therefore, you can get your answers as they are. GGU according to the base pairing policy and principle. That's how it works. So that's the sequence class. And then the protein that DNA molecule codes for is made up of 50 amino acids. How many of each of the following is involved in the formation of these proteins? We are saying it is made up of 50 amino acids. We are saying the protein that this DNA molecule codes for is made up of 50 amino acids. How many of each of the following is involved in the formation of this protein? And the RNA nucleotides. So you go again, you should see that this one is also given according to the formula that I was giving you on Metric Hub. Again, it's just a repetition of the same questions that we are working again and again. So, class, I want you to appreciate, and also I want to appreciate you, and thank you so much for your time. And I hope that you also have the best interest and the best time over Metric Hub and all your exams. For those who are following Metric Hub, I don't want to explain anything or market anything. I'm sure you can see all this paper. It is just a duplicate of quite a lot of work that we have worked there and explanation that we have given with reference to say how do we work out these questions. We have worked them quite a lot of times, and they are basically repetitively coming. So for those who love their NSFAs and who are working out to ensure that next year something better is coming, please connect to Metric Hub and share to friends and family. Subscribe and join membership for the best of your time. And again, I promise you, you will never miss and fail anything. All the best. Let's meet again in the next term work and every other piece of work coming. Thank you.